So hello everyone. I think we can just start the session for today and let everyone join afterwards. Okay, so good evening everyone. Welcome to the week 7 section on the NPTEL course on cell biology, cell organization, division and process. So my name is Isaac Cherry and I am a 50th PhD scholar at School of Life Science, JNU Delhi. So we will be discussing about today about 30 questions, 10 from the current NPTEL section for this course and 10 from the previous year NPTEL section and 10 questions that I just made. So I think we can just start the session for today. So I hope I am visible and I am clearly audible also. So coming on to the next first question. Which of these pathways sends levels of NAD plus and regulates aging? mTOR, IGF-1, sirtuins and AMPK. Which might be the answer? Which might of these pathways sends level of NAD plus and regulates aging? If any one of you know, just type in the chat box or you can unmute yourself and give the answer. No issue in the both case. Okay, so the answer to this question is sirtuins, option number 3. So basically, if you look into other factors like you can see that this diagram here, you can discuss that afterwards. So if you look into this answer, like this sirtuins actually sends the level of NAD plus in the body, and it has the capability to regulate the whole like the aging pathway. You can see that in case of dietary restriction, whenever dietary changes are there, the levels of this energy producing source also changes in accordingly. So sirtuins actually recognize this particular levels of NAD plus, and it activates this particular enzyme phosphoglyceride cyclase 1 alpha, and it inhibits aging. So the levels of NAD plus actually determines whether the cell has to what you say the cell has to actually undergo aging or not. When it comes to the other options like mTOR, you can see that it comes in the downstream pathway, right? Majority of the pathways actually coincide with or correlate with mTOR kinase. This is a kinase actually for release certain proteins and all. So it actually promotes aging as compared to sirtuin. When sirtuin levels are high, it actually inhibits aging, while mTOR levels are actually high, it actually promotes aging. Similarly, if you look into the other factors like IGF-1, and in case of mTOR, it senses high amino acid concentration also. It also regulates with the like independently regulate with other factors that are involved in the pathway, but independently it has the ability to regulate or sense high amino acid concentration. And also IGF-1 is another protein that is involved in this case. It is a growth factor. It actually acts in the other presence of other kinase like phosphatidylinositol 3 kinase and also AKT kinase in this case, and it interacts with the mTOR and also inhibits other pathways like FOXO proteins and all, and thereby regulating aging. You don't have to go detailed into it, you just have to look into pathway and all. And AMP kinase also, from the name itself, AMP kinase senses the level of AMP in this case, which is also a high energy compound, right? So in that case also it senses this level and then inhibits the mTOR kinase, which then reduces the aging level and all. So each and every pathway, you can see that each and every protein in this particular pathway has the capability to sense each and every different compounds including IGF-1 which senses glucose and growth hormones, presso growth hormones and all, and mTOR which senses high amino acid concentration, and AMP kinase which senses the AMP level, and also as we have discussed here the sirtuins which senses the level of NAD plus, and thereby all of them correlate with each other and also independently, and then affects the whole aging process in the body itself. So yeah, so the answer to this question is sirtuins which senses the level of NAD plus and regulates aging. So anyone has any doubt regarding this? Okay, so I think I'm clear. So I hope that I will just move into the next slide then. So coming on to the next question. Rapamycin, which is a drug, inhibits which of these enzyme complex? mTOR kinase, AMP kinase, glucokinase, or none of the above? Which might be the answer? Out of which of this just rapamycin inhibits? Yeah, I'm getting option number 1. Any other option? Anyone has any other answer to this, response to this question? Okay, so the answer to this question is you are right. It is mTOR. Rapamycin inhibits mTOR kinase. So basically rapamycin is actually an inhibitor of autophagy, as inhibition of mTOR mimics cell starvation by blocking the signals required for cell growth and proliferation. So you can see that in the previous slide we can discuss this, right? So mTOR, so it has multiple functions. It has capability to regulate aging as well as it has a capability to regulate autophagy also. So in this case it inhibits the mTOR kinase that is present here. So it indirectly inhibits the mTOR kinase, not directly, by blocking the signals required for cell growth and proliferation. What it does is that it binds to this particular protein, which is called as a FK506 binding protein, which is a 12 kilodalton protein. The molecular weight is about 12 kilodalton. And this protein consists of this particular binding domain because it has the capability of binding this as analog, and rapamycin is actually an analog for this particular binding protein there. So when it binds to this protein, what it does is that it inhibits this particular activity. So this is the complex that rapamycin mTOR forms with other protein complex including LSD8 and RAPTA, forming mTOR complex 1 or mTOR C1 in this case. So whenever this so what happens is that whenever rapamycin binds to this particular protein, FK506 binding protein or FKB12, which is the 12 stands for the molecular weight of this protein, it inhibits this complex formation there. So what happens is that whenever it inhibits the complex formation, it promotes the autophagy, because mTOR is actually an inhibitor of autophagy. It in the previous case it promotes aging, but in this case it inhibits autophagy. So that happens in this case. So whenever mTOR complex is not formed there, the autophagy will start to happen basically, while the nucleotide lipid protein synthesis that has been going on or keeping the cell intact or cell survival intact stops there. It gets inhibited there. So the whole pathway goes into opposite mode in this case, because whenever the mTOR is active, it actually inhibits autophagy and it promotes the nucleotide lipid and protein synthesis. But when rapamycin bound to this particular protein FKB12, it inhibits this complex formation, and therefore it promotes autophagy and inhibits the synthesis of nucleotide lipids and protein. So basically it happens through this particular pathway, right? You can see that the growth factors including the insulin and also the insulin actually binds to this particular inhibitor. It activates this phosphatidylinositol 3 kinase and AKT kinase in this case, and then it actually promotes this pathway. So rapamycin inhibits this particular mTOR kinase and thereby promotes autophagy in this case. So whenever the activity is that basically it's an analog of this particular FKBP12 protein. So I hope I am like it's understandable. So anyone has any doubt? Okay, so I hope it's clear. So I will just move on to the next question. So coming on to the next question, cell swelling, membrane rupture and inflammation can accompany which of this process? Apoptosis, necrosis, senescence, or all of the above? Which may be the answer? So I will repeat my question again. I think there was a connection issue. Cell swelling, membrane rupture, inflammation can accompany which of the following option? Apoptosis, necrosis, senescence, or all of the above? Anyone? Okay, I am getting option number 2. Okay, so the answer to this question is option number 2, necrosis. You are right. So basically, if you look into necrosis and apoptosis, so basically this is the diagram. So first we will go with the necrosis because that's we are dealing. So it is a form of a premature tissue death that occurs due to various injuries or diseases, contrasting with the programmed cell death. Is similar to apoptosis, but not in comparison to that. There are several differences in this case. So it involves uncontrolled cell death, leading to cell swelling, organelle disruption, and eventual cell lysis. And due to factors like these factors, that is cuts, burns, bruises, oxygen deprivation, which is also called anoxia, and hypothermia can be one of factors. Other factors include immunological attacks, infection and disease, and also conditions like avascular necrosis or gangrene, which means like the cell death is happening there. So necrosis involves early cellular changes like mitochondrial swelling, genetic material condensation, and eventual cell membrane rupture. This can lead to inflammation and tissue damage. So these are the secondary process that take place in necrosis, and these are the primary process. Starts with this primary process and then leads to the secondary process and all, like inflammation and tissue damage. So if you look into the difference between apoptosis and necrosis, these are the two differences. So in case of necrosis, you can see that this normal cell. So what happens is that in case of there is any like these conditions, including the infections are there, if any diseases are there, or any cuts or burns or happen within the body near to a particular cell, what happens is that the cell starts to form more, undergo blubbing and all. The structure of the nucleus changes. So it is affecting the nuclear membrane there. So the structure of the nucleus actually starts to begins to change. and these blobs start to form within the cell. You can see that the cell is getting regular in shape and all from this primary architecture and all. And these blobs then starts to fuse together and becomes larger. No organelles are located in the blobs. So in case of apoptosis, it's a totally different thing. Organelles are actually involved there. And then when these blobs are starting to becoming larger, the cell big membrane actually ruptures and releases the cell contents, and the organelles are non-functional in this case because the cell is no longer viable there. So it starts to release out its contents because of the fusion of these blobs that are forming within the cell, and also the nuclear integrity is getting affected there, and the organelle starts to go out of the cell. So that's how the cell rupture in case of necrosis. But in case of apoptosis, what is that? These blobs actually forms in this case, and the nucleus begins to like break apart, and also the DNA breaks into small pieces. So DNA damage also take place in this case. Once the nuclear envelope starts to break down, the DNA strands within the nucleus also starts to break down into small pieces. That means that DNA fragmentation is followed by nuclear membrane breakdown is followed by the DNA fragmentation that is located in this case. And organelles are also located in the blood. So you can see that within the bloods that are present in the necrosis, you can see that there was no organelles that are involved in this case. But in this case, in case of apoptosis, inside the bloods, the organelles can also be found there. And then afterwards, in this case, necrosis, the cell ruptures and the contents are released out, right? In case of apoptosis, what happens is that the cells break into several apoptotic bodies, and the organelles are still functional in these apoptotic bodies there. So that's how apoptosis happens. In this case, you can see there are considerable difference in this case as compared to apoptosis. And one other thing is that in case of necrosis, the process can be painful because of infection and or any other cuts that are happening. It is painful. It is because of that it is happening, right? But apoptosis is also called as programmed cell death. It's what which is present within the cell, right? It is pre-programmed within the cell that if the cell has to undergo this process, then apoptosis can happen, leading to cell death and all. So it is quite painless. It happens like majority of the time in our body and all. But necrosis can be due to majority of the case some external factors. Some internal factors can also be part, but majority of the time it is due to external factors in this case. So that's the difference between apoptosis and necrosis. Senescence we will also discuss, but it is in a kind of a process where the cell undergoes some cell arrest or cell resting phase, and all. We will discuss that afterwards. So up to this point, I think it is clear. So anyone has any doubt? If anyone has any doubt, you can type in the chat box, no issue. Okay, so I think I am clear. So I will just move on to the next question then. Coming on to the next question. Which of the following is a pro-apoptotic gene? BCL2, MCL1, BAC or A1? Which is the answer? Which of the following is a pro-apoptotic gene? Okay, so I will just give you the answer. So the answer to this question is BAK or BAC. So several proteins are there, right? So some of them are caspases, some of them are kinase, etc. So basically what happens is that there are several category of different apoptotic protein. Okay, it can be either pro-apoptotic, it can be anti-apoptotic, and pro-apoptotic can be either be like this, what is it, BH3 only proteins or multi-domain effector proteins. So if you look into the first factor, see these are some of them are given here, some of them are given in different factors, right? So you can see that in case of pro-apoptotic proteins, BH that it contains only BH3 domain, you can see that BAD, BMF or BIM, a bit protein, these are all there. So some of them you have heard, some of them you might not have heard at all. And as compared to when what happens with protease, these proteins have the capability to inhibit this anti-apoptotic proteins, which includes BCL2, which can be of different family proteins, that is XL can be there or W can be there, and MCL1 that is described here, and also A1 proteins. So these BCL2, MCL1 and A1 proteins are anti-apoptotic protein. They are also called as guardian proteins, which prevents the apoptosis of cells. And these are inhibited by this pro-apoptotic proteins consisting only BH3 only proteins, which are also called as sensor proteins. You can see the example is BAT, BIT, BIM, etc. Open is also there. Yeah. So this inhibits this anti-apoptotic proteins or guardian proteins. And the afterwards, what comes in another category of we have discussed before, right? Another category of pro-apoptotic protein is multi-domain effector proteins. So these are also called as effector proteins there. So because it comes in the downstream pathway of the apoptosis pathway, and it directly regulates the apoptosis and everything, as compared to the other upstream proteins that are involved, that can be either be pro-apoptotic or anti-apoptotic in nature. So this BAX and BAC, you can see that this one protein is given there. They are also called as pro-apoptotic multi-domain effector protein. So these are under the regulation of these upstream proteins that are found here, or it can directly be regulated by this these particular proteins that are given in the center of this diagram there, that is BIM or BIT protein, that can actually increase the level of BAX or BAC, promoting apoptosis, while the anti-apoptotic proteins like BCL protein, MCL or A1 protein can inhibit the BAX or BAC protein forming the, which inhibits the pore forming complex of capability of BAX or BAC, because it can form pores at the intermembrane surface, right, mitochondrial surface and all, and thereby promoting apoptosis. So in this protein, the pro-apoptotic proteins, that is the proteins which promote apoptosis, include BAC. All the other proteins are anti-apoptotic proteins, or also called as guardian proteins. So yeah, BAC is the pro-apoptotic protein gene that is found here, and it is written italicized because it is actually a gene. If it is given as protein, it can be straightened out, or it can be, it starts the capital letter and followed by a small letter there. So I hope this is clear. So anyone has any doubt? Anyone has any doubt? Okay, so I think you have understood the topic. So I will just move on to the next question then. Coming on to the next question. Which of these options which are given below is a homologue of set 4 in mammals? APAF1, caspase 3, BCL2 or BAX? Which might be the answer? And this is actually present in Habitus elegans, cell death 4 protein. It has a homologue in mammals. So out of which these options, which is that particular homologue? Okay, so I think I will just give you the answer. So answer to this question is APAF1. So basically this is the small diagram. This you might have seen it in the lecture videos and all. So this is happening in the C. elegans, this pathway, and this is happening in mammals. So in case of C. elegans, you can see that Z4 or cell death protein 4 is interacting with this Z3 and then promoting apoptosis. So this whole pathway is placed in C. elegans, and it is inhibited by these two proteins, and this pathway is interpathway is inhibited by this Z9 protein. In case of mammals, this Z9 correlates with the BCL2 family proteins, which is actually preventing apoptosis, right? It's an anti-apoptotic protein. So APAF1 interacts with other caspase like caspase 3 and 9 and then promotes apoptosis in this case. So in this case, if you compare the diagrams, you can see that Z9 is actually a homologue of BCL2, Z4 is actually a homologue of APAF1, and Z3 is actually a homologue of other caspases that like caspase 3, etc. and all that promotes apoptosis. So even in case of different animals which have been studied, in case of C. elegans it was first discovered, right? So B cell family proteins was first discovered. So if B cell family of protein was first discovered, the correct nomenclature that was given at that time was Z9 to that. So you can see that it is homology is considerable there, and it has to be considered at different points of each level of organization, higher to lower eukaryotes or higher to level organisms and all. So the answer to this question is APAF1. So we will discuss with the APAF1 in the some of the other questions that we will be dealing with. So up to this protein, you can see that, and caspase 3 is also a inhibitor of initiator of apoptosis, etc. And BCL2, as we have discussed, is an anti-apoptotic protein, and BAX or BAC is actually a pore-forming protein which promotes apoptosis within the mitochondria. Yeah, so APAF1 is also a promoter of apoptosis. Only one is an anti-apoptotic, which is BCL2. Others are pro-apoptotic in nature. So I hope this is clear. So anyone has any doubt? Okay, so I think it is understood. So I will just move on to the next question. So coming on to the next question. Which of the following ion plays a crucial role in the control of various forms of cell death, that is necropsy, apoptosis and autophagy? Calcium, chloride, potassium or oxygen? Which may be the answer? Which of these ion actually regulates cell death process and all? Okay, so I think I will just give you the answer. So the answer to this question is option number 1, calcium. So basically calcium ions, or it is a divalent cation, right? So that's why it's represented as 2 plus. Here also 2 plus is there, and are involved in signaling pathway that leads to apoptosis. An increase in intracellular calcium triggers release of pro-apoptotic protein from the mitochondria, such as cytochrome C. Cytochrome C is actually important trigger for this apoptosis, which activates caspase and leads to cell death. The flux of calcium from the endoplasmic reticulum to the mitochondria is critical for initiating apoptotic signal, and it can lead to opening of mitochondrial permeability transition pore, causing mitochondrial swelling and rupture, which further promotes apoptosis. Several of the pore forming complexes are actually produced by these two proteins. This protein, like which is pro-apoptotic nature, that is BAX and BAC that you discussed earlier, right? So the BCL2 family proteins actually more like BCL2 as well as which is a anti-apoptotic protein, and Bax, which belongs to a pro-apoptotic protein, modulates the calcium homeostasis and influence the apoptotic pathway. They both have the capability of regulating the apoptosis pathway and all. BCL2 can inhibit the apoptosis by reducing calcium transfer from ER to mitochondria, while Bax promotes cell death by increasing its transfer from the opposite, that is ER to mitochondria itself. So you can say that in the normal pathway, when the cell is living, the IP3 generating stimuli actually helps in the mitochondrial calcium uptake, but up to a certain level. So it is actually transient in nature, so it does not accumulate in this case. So it stimulates the organelle metabolism, and where electron transport chain actually happens, followed by ATP synthesis activity, ATP production actually happens in this case. So this is the pathway that is happening in normal cell when the cell will survive. But when apoptosis need to happen, what is that? Several other extrinsic pathway as well as intrinsic signal molecules also take place in this case. This leads to excessive increase in calcium level in this case. So you can see that the BCL2 and Bax also regulate this pathway. Bax actually increase the level of ER calcium, and the inositol 3-phosphate receptor is actually present at cell surface, actually helps in the uptake of calcium into the mitochondria from the ER. So basically this receptor is present in the ER, right? So through this receptor, through this transporter protein, that it has the capability to reach mitochondria. So once the calcium has reached mitochondria, it starts accumulating in this case. When it accumulates, it leads to overload stage, and which promotes the release of caspase cofactors like caspase 3, caspase 9, etc. And then once this level is increased up to a particular level, the apoptosis can happen and cell death pathway is activated in this case. So this is how the calcium actually plays an important role in regulating the cell death pathway, any of the pathway, necrosis, apoptosis, or autophagy. So its overload can lead to the activation of caspase, which promotes apoptosis. And the transfer of calcium actually takes place from the ER to mitochondria based on some regulation of these pathways like proteins like BCL2, which is a anti-apoptotic protein, and Bax, which is a pro-apoptotic protein. So that's how the whole pathway is regulated in this case. So I hope I'm clear. So calcium ions play a crucial role in the control of various forms of cell death, that is necrosis, apoptosis, and autophagy. So does anyone has any doubt in this case? I hope I'm clear. Okay, so I think it is understood. So I will just move on to the next question. So coming on to the next question. PET scan uses which of the following properties of cancer cell? Cancer cell import large quantities of glucose. Cancer cell import large quantities of ATP. Cancer cell imports large quantity of sodium ions. All of the above. It is a method that is used to detect whether cancer is present in the body and all, right. So what is this principle based on? Yeah, I'm getting option number 1. Any other option? Yeah, so basically you are right. Cancer cells import large amount of quantities of glucose, right? So PET scan is also called, the full form is positron emission tomography. So it is primarily uses the property of cancer cells importing large quantities of glucose. This is because cancer cells metabolize glucose at large size. It has the capability to undergo large amount of glycolysis, and then it undergoes fermentation and all, leading to the formation of lactate and all. So this is, instead of going into oxidative phosphorylation and all, it undergoes this lactic acid fermentation pathway, leading to the formation of lactate and all. And this lactate is actually used up to build most of the components that are required to maintain this cancerous cell and all, as compared to a normal cell where ATP is produced by oxidative phosphorylation and other pathways. Much more other components or the survival of the cell depends on the oxidative phosphorylation. Cancer cells actually rely on lactic acid fermentation and everything. So that's what Warburg effect means actually. It depends on the aerobic glycolysis process leading to utilization or metabolism of higher number of glucose as compared to normal cells in a much higher rate. And this increased glucose uptake is detected by PET scan using radio tracers like this particular 18 fluorodeoxyglucose and all, which accumulate in the areas of high molecular activity such as tumors. So it has the capability to undergo large amount of metabolize large amount of glucose, right? So if you use this particular radio tracers like this fluorinated deoxyglucose and all, you can see the accumulation of this particular intermediate within the tissue of a part of our body where it is getting higher amount of glucose is getting uptaken there, because it is actually a kind of a glucose analog, so it is also getting uptaken in the body. However, as compared to a normal glucose, this has the capability to emit some amount of radioactivity there. So based on the radioactivity counts that are present within our specific region of the body, the PET or positron emission tomography actually detects where this is getting accumulated and suggests that this and from the image that you are getting, we can suggest that this particular part of the body might be cancerous in nature. So yeah, so that's how a PET scan actually works. So PET scan uses the property of the cancer cell to import large quantities of glucose into the body in order to provide subsequent detection and everything. So I hope I am clear. So anyone has any doubt? Okay, so I think it's clear. So I will just move on to the next question. 1 minute. Okay. So coming on to the next question. Which of the following is not a hallmark for cancer cell? Resisting cell death, enabled replicative mortality, genome instability, none of the above. Which of the following is not a hallmark of cancer cell? Some of these properties are possessed by cancer cell while others are not, which may be the option. Okay, I am getting option number 2, replicative immortality. Any other option? Okay, so the answer to this question is none of the above. So all the other is actually a hallmark of cancer cell. It can resist cell death. It can undergo replicative mortality because replication actually happens within the cell. So only after replication continuously happen, continuous cell division can happen, right? So cancer cells have the property to divide continuously for a long period of time. So in order to promote that, the replication should also be continuous there. So it has the capability to undergo replication immortality. It has the capability of replicative mortality there. Genome instability is also there that promotes replicative immortality and lead to higher cell division, which resists because of that it has a probability of resisting cell death. So if you look into the option, the answer to this question is none of the above. So I hope I am clear. So anyone has any doubt? So option number 2 is actually a hallmark of cancer cell. So here none of the all these three are actually a hallmark. Anyone has any doubt? Okay, so I hope this answer is clear. So I will just move on to the next question. Okay, a state of stable cell cycle arrest in response to diverse stress or intercellular damage is termed as which of these options? Cell death, senescence, aging, or none of the above? A state of stable cell cycle arrest. Out of which of these options which might be the right answer? Okay, so I think I will just give you the answer. So answer to this question is senescence. It's a basic question, right? We have discussed this before also. So basically, cellular senescence is characterized by a stable and generally irreversible cell cycle arrest, which occurs in response to various intrinsic and extrinsic factors such as DNA damage, oxidative stress, and oncogenic activation, or any other process, etc. And this state prevents the proliferation of damaged cell, acting as protective mechanism against potential tumorigenesis. So, as compared to other factors like autophagy, necrosis, necrotopsis, or this apoptosis, senescence actually does not lead to cell death. It prevents the proliferation of damaged cell. What happens is that it promotes a cell cycle arrest at a particular cell cycle checkpoint. So the cell cannot move across this checkpoint. It stays within that particular checkpoint or within that particular phase of the cell cycle, so that it does not undergo cell division and all. So it cannot move into the M phase. It is actually stuck within that phase. So as compared to apoptosis, where the cell death happens, cell death does not happen here. It actually arrests the cell at that particular phase of the cell cycle. So that's what happens. So a state of stable cell cycle arrest in response to diverse stresses or intracellular damage is termed as senescence. So I hope this is clear. So this is a basic one. However, if you have any doubt, you can just ask now. Okay, so I think this is a basic question, right? I think it's quite understandable. So I will just move on to next question. Coming on to the next question, which of these caspase have a CAR-D domain? Caspase 2 and 8, caspase 2 and 9, caspase 8 and 10, or caspase 8 and 9? Which may be the answer? There are several caspases. Some of them have this CAR-D domain, some of them have a dead domain. Out of this caspase that are given in the option, which of those have the CAR-D domain? Okay, I'm getting option number 2. Anyone has any other answer or response? So basically you're right, option number 2 is the correct answer. That is caspase 2 and 9 has CAR-D domain. So basically these are called as initiator caspases. The execution of caspase like caspase 3, 6 and 7 doesn't have this particular domain there. You can see that there are only two domains, L domain and S domain there. As compared to initiator caspases, there are also two subtypes, one containing the CAR-D domain and other containing the dead domain. So CAR-D domain is also called as far as you recall, it is caspase activation and recruitment domain. And the DED is also called as death effector domain. Like, please check it out. I am not sure whether I am right or not. So basically, caspase 2 and 9 has this CAR-D domain, and caspase 8 and 10 has this dead domain. So what it does is that it has a capability to bind to certain receptors that are present in the cell surface. So it is mainly involved in this extrinsic pathway of apoptosis. So this caspase 2 and 9, with this particular domain, has the capability to bind to this particular receptor that are present in the cell surface and initiate the downstream pathways or apoptosis there, including the activation of caspase 3 and caspase 3, etc., which can lead to apoptosis and everything. So yeah, so here caspase 2 and 9 contains the CAR-D domain, while caspase 8 and caspase 10 consists of this dead domain or DD domain. And other caspases that play in downstream pathways called as execution caspases does not have both of these domains. It consists of only these two domains there. And these two domains are conserved, highly conserved in all the caspase types. So I hope I am clear. So anyone has any doubt regarding this? Okay, so I think it's clear. So I will just move on to the next question. Coming on to the next question, which pick the correct order of steps in performing the 3C technique. Crosslink, you can read the option: crosslink, restriction diation of chromatin, ligate, purify pair entrins, crosslink, ligate, restriction diation, purify or pair entrins, crosslink, ligate, purify, restriction diation and pair entrins, and crosslink, purify, restriction diation, ligate and paired entrins. Which might be the answer? 3C is actually a technique that is used to study, so it has a specific step that is involved there. So which might be of this option might be the right answer. It starts with cross-linking and ends with paired entries. In between it might change based on the option which is the right option. Okay, so the answer to this question is option number 1, that is the cross-link followed by restriction diation of chromatin, ligation, purification and paired entries. So this is the whole step. So what 3C stands for is 3, it is actually, sorry, okay. So it's actually also called as 3D conformation, chromatin conformation analysis technique. So whenever in case of a chromatin, there are chances that the two DNA sequences can interact with each other. It can be either from a homologous chromosome or it can be from different chromosomes, right? So what happens is that in this process we have to study the interaction between two DNA sequences that are happening within the chromosome or two different chromosomes in the chromatin, three-dimensional chromatin structural organization and all. So what happens is that we actually introduce a cross-linking agent, including like examples including formaldehyde. What it happens is that it cross-links the two DNA sequences that are present in chromosomes together. You can see the cross-linking here. Afterwards, the other part of the chromosomes are getting degraded by using specific any kind of enzymes or any kind of in this case restriction diation is given, or any other kind of shearing methods, etc. So what happens is that all the other DNA gets degraded and the DNA fragments that are cross-linked will stay cross-linked to each other, and then they are ligated to each other so that the DNA ends getting is attached to each other. Once they are ligated, they are purified from the cross-link. They are purified and then they are linearized. So in this case, like you can see that this is a normal genomic PCR conformation and all. In case what happens in 3PCR is that we actually generate different kinds of primers there. Primers that can bind to different regions of chromosomes there. Or if we have a specific interest in studying a specific DNA sequence, we can actually get the DNA sequence and generate primers accordingly to that. So accordingly primers are generated. You can see that the forward primer is there and the reverse primer is there. Forward primer will be accordingly to one of the fragments, and the reverse primer will be to the other fragment that might be interacting with each other. And afterwards you do a PCR accordingly, and then visualize the band in a genome DNA PCR. You can see the band here. So based on that, if the band is appearing or not, you can suggest that actually these two, if the band is present there at the appropriate size that we are actually determining, then we can say that they are interacting together in the chromatin architecture and all. If they are not band there, then it means that they are not interacting with each other. So the whole process of the 3C is actually dictated there. It starts with the cross-linking, then restriction diation, then you are, in order to make a paired end read and all, you are ligating it out, and then you are purifying it and then linearizing it, and then doing a PCR or a paired end read using the specific primase accordingly, and then you are visualizing it in a gel. So from that, if you are getting a band, they are interacting. If they are getting a band, it is not interacting. So that's how the whole 3C conformational technique goes. So I hope it is clear. So anyone has any doubt? Okay, so I think there are no doubts. I will just move on to the next question then. Okay, so coming on to the next question. The process of spreading of tumor from the site of origin to the other site of the body through the lymphatic or a circulatory system is termed as which of these options? Benign, malignant, metastasis, or all of the above? The process of spreading of tumors from site of origin to other site of the body through a circulatory system. Okay, I am getting option number 3 as well as option number 2. So I think I will just give you the answer. So the answer to this question is metastasis. We are actually asking about the process where the tumor spreads, right? So if you look into option number 1, benign means that the tumor is not spreading, it's actually present within that particular site of the body. It can neither be malignant and neither it can undergo, because of that, neither it can undergo metastasis. So yeah, normal moles that are appearing in the body are kind of benign in nature. It cannot spread to other part of the body, right? As compared to malignant, malignant means that it's actually a kind of a cancerous growth there. It has the capability to spread within that particular region and also has the capability to undergo metastasis. Metastasis means that it can move from that particular part of the body to the other part of the body. So the third option is actually the process there. Malignancy means that the tumor can spread. It can spread within that site itself or to other site itself. But the whole process of spreading of this malignant tumor from one part of the body to the other part of the body, anywhere where it can attach and then proliferate to become a cancerous growth there also, that particular process or phenomenon is called as a metastasis. So malignancy is actually a carcass of like this cancerous tissue. That means that it can spread. It can spread either at that position or to other parts of body. But the process actually is designated by this term metastasis, that it can spread to other part of the body from its original site. So that's called as metastasis. So the answer to this question is metastasis, not malignant. So malignant is kind of a broader term. Metastasis actually dictates that particular process of spreading from one part to another. So I hope I am clear. So anyone has any doubt? Okay, so I think it is quite clear. So as there are no doubts, I will just move on to the next question then. Okay, coming on to the next question. Which of the following virus can cause cancer? Papilloma virus, HBV virus, or hepatitis B virus, EBV virus, or Epstein-Barr virus, or all of the above? Which may be the answer? Which of these viruses can cause cancer? So the answer to this question is all of the above, option number 4. So human papilloma virus, the first option, are known to causes these kinds of cancer: cervical, anal, penile, as well as oropharyngeal cancer. HBV or hepatitis B virus is associated with mostly liver cancer, right? Particularly hepatocellular carcinoma. Epstein-Barr virus is linked to various cancers like Burkitt's lymphoma, Hodgkin's lymphoma, and nasopharyngeal carcinoma, which happens within the throat and all. So all of these viruses actually causes cancer. So anyone has any doubt? Okay, so I think it's clear. As there are no doubts, I will just move on to the next question. Okay, this is a basic question. Which process involves programmed cell death essential for development and homeostasis? Apoptosis, necrosis, autophagy, and senescence. Yeah, option number 1, apoptosis, right? So apoptosis is also called as programmed cell death, and especially in case of some nervous tissues and all, apoptosis is common because at a certain cell point the nerve cells has to get degraded down so that the particular architecture of nervous tissue has to be maintained, so they can undergo programmed cell death. Some of the cancer cells also undergo programmed cell death. While necrosis is not a programmed cell death, it can be due to several external factors. What happens to a cell, including injury, etc., that we have discussed before, or any kind of cuts or any burns that are happening within the body, or any kind of something that leads to inflammation, eventually leads to cell death and all. Autophagy is a process by which the cell starts to devour itself. So it is also similar to apoptosis, but the mechanism of autophagy is quite different from each other. And at this point, it is not essential to discuss autophagy at this stage. And senescence is the stage at which the cell arrest actually happens. Whenever the cell, any kind of replicative damage or anything happens, instead the cells does not need to undergo apoptosis. They enter into a cell cycle arrest stage where they no longer enter the cell division or the mitotic phase, and it gets arrested into a stage without the cell death actually happening. So I hope it is clear. So anyone has any doubt? Okay, so I think it's quite clear. So I will just move on to the next question. Coming on to the next question. Which of these disease is not caused due to excessive apoptosis? AIDS or HIV, neurodegenerative disease like Alzheimer's or Parkinson's, cancer, stroke. Which may be the answer? Which of the disease is not caused due to excessive apoptosis? I am getting option number 2, not caused due to excessive apoptosis. Yeah, so basically you are right. The answer to this question is neurodegenerative disorders, right? So in case of like HIV and all, you can see that yeah, basically you are right, neurodegenerative disorder. So in case of apoptosis, you can see that in case of HIV, you're right, so which is human immunodeficiency virus, right? So what happens is that much of the macrophages and everything gets degraded out, right? So basically, when HIV infects, this virus infects a macrophage or any other immune cell, what happens is that it promotes the cell, the virus actually gets replicated within the cell, and then it promotes the cell to undergo apoptosis. So because of excessive apoptosis that is happening within the cell, the count of immune cells goes down and HIV begins to thrive within the body. In case of cancer, you can see that cancer has the capability to undergo continuous division, right? Because it has the capability to undergo continuous proliferation because of the reduced apoptosis that is happening within the cell. So that's how the cancer cells thrives and grows. In case of growth, at one part of the body, the neurons start to degenerate, right? So it is because of excessive apoptosis that is happening there, and the one part of the body becomes partially paralyzed and all because of that neurodegeneration they are happening there. So in case of neurodegenerative diseases, it is not because of apoptosis. Some of the other factors like protein accumulation taking place there that actually leads to this particular process there. So that's the answer, neurodegenerative diseases. So anyone has any doubt regarding this? Sorry, I would like to yeah, so answer to is right. So I hope that I am clear, so I will just move on to the next question. So coming on to the next question, what is the primary characteristics of cellular senescence? Uncontrollable cell death, cell division, enhanced metabolic activity, permanent cell cycle arrest, increased telomerase activity. Which of these is a primary characteristics of cellular senescence? Yeah, I'm getting option number 3, permanent cell cycle arrest. Yeah, so the basic answer, right? So you know the answer, permanent cell cycle arrest. There we have discussed this before. And some of the main triggers of cellular senescence I have given here. So this can be a resultant of oxidative stress, genotoxic stress, or ionizing radiation. So persistent DNA damage response can also lead to senescence. And also another process is replicative senescence, where it happens when the telomere shortens to a critical length, triggering DNA damage response that halts the cell division. So this particular process is called as replicative senescence because of the shortening of telomere to a particular length. Instead of undergoing apoptosis, it undergoes replicative senescence there. Activation of oncogenes leading to oncogene-induced senescence can also be another process, and it can be a protective mechanism against cancer. And other increased levels of RO species or reactive oxygen species can also induce senescence there. And other functions include mitochondrial dysfunction and nutrient deprivation. So and also chronic inflammation and also tissue damage signals can also induce senescence in the affected cells. So these are some of the other factors that can lead to senescence. So it is some of additional knowledge that I am giving here. So I hope it is clear. So anyone has any doubt? Okay, so I think it's clear. So I will just move on to the next question then. So coming on to the next question, what is Hayflick limit? The number of times DNA replication happens in the cell, number of times a cell divides before it stops dividing, number of times a cell can be passaged before it becomes inviable, or none of the above. What is Hayflick limit? Okay, so I will give you the answer. So the answer to this question is not option number 3. So the answer to this question is option number 2. The number of times a cell divides before it stops dividing. So basically, whenever a cell divides, right, it is apart from a cancerous cell, consider a normal cell there. It can divide up to a particular limit. and afterwards several reactive oxygen species gets incorporated into a cell. It starts to get accumulated. The telomere starts to get shortened, and other factors that leads to that promote cell death. So up to a particular point, the cell can divide, and afterwards, whenever these factors like telomere shortening and everything happen, the cells start to stop dividing, or it starts to undergo some chromosomal changes, like it can undergo euploidy, aneuploidy, etc. Ploidy changes happen within the cell that renders the cell inviable in this case. So whenever it happens, the cell stops dividing after that particular certain point of time. So there is a time limit about which the number of times a cell actually divides before it stops dividing there. So that particular uh, definition is governed by this term Hayflick limit there. So it means that the Hayflick limit refers to a number of times a cell divides before it stops dividing. So if you look into the third option, like it's a different thing, something related to a cell culture there. The number of times a cell can be passaged before it becomes viable. So because it's also similarly, but directly if you consider the definition of Hayflick, this is the second option. So passaging means that whenever you are doing a cell culture, right? So there's a point where the cell starts to accumulate the cell culture dish, about which the cells start to die off. So what you need to do is that you have to take some amount of this culture where the cells have divided, and then you have to inoculate it into a new culture, different culture or culture plate with a new media there, because in the previous media the nutrients and everything start to deplete out. Majority of the cell culture plate is occupied by cell, it is crowded there. So when you are inoculating that particular culture, some amount of this culture into a new plate or a culture plate, the new media, the cells start to grow itself, basically that. So that the cells can be like passage up to a particular time, because what happens after it has crossed a particular limit, the telomere, as we have discussed here, the telomere length shortens and everything, and then it cannot be passage anymore. You need a new batch of cells to undergo your experimentation and everything. So if you refer to Hayflick limit, the second option is the right one, that is the number of times a cell divides before it stops dividing. So I hope I am clear. So anyone has any doubt? Okay, so I think there are no doubts. So I will just move on to the next question then. Coming on to the next question. The accumulation of which type of cellular damage is a hallmark of aging? DNA mutation, protein misfolding, lipid peroxidation, and all of the above. Yeah, I am getting option number 4, all of the above. Any other option? Also getting option number 2. So the correct answer to this question is option number 4, all of the above. So basically DNA mutation accumulate in the aging because all the protein and the cell function is getting affected there, right? So mutations start to accumulate, the cells start to die off. Protein misfolding is also a cause in case of neurodegenerative disease and all. Lipid peroxidation is also happening in this case, because lipid peroxidation actually degrades the lipids that are present within the cell there, that are stored within the cell. Therefore they cannot be utilized there, or some of the lipids that lipids are actually essential for maintaining some of the functions, right? Example for fatty GP angle protein and all, they have a lipid angle everything, right? So if the lipid peroxidation happens, they get degraded, and it also is a common factor of aging, because all the metabolic process that are happening in the cell is undergoing a considerable change, or the metabolic activity is getting reduced there because of protein changes and everything, or DNA mutations happening there. So the answer to this question is all of the above. So anyone has any doubt? Okay, so I think it's quite clear, so there are no doubts. So I will just move on to the next question. Okay, coming on to the next question. Which protein is a key regulator of apoptosis in the intrinsic pathway? P53, BCL2, TNF alpha, or caspase 1? Okay, I'm getting option number 2. Any other option? Okay, so BCL2 is the right answer, so you're right. So basically, this is a intrinsic pathway and everything, right? So you can see that several proteins are there. And also in this case you can see that in case of extrinsic pathway you can see that several receptors are there, TNF receptor are there, FASL receptors are there. So these are also receptors that recognize this particular extrinsic stimuli that that tells the cell that you have to undergo apoptosis. So you can see that there are several domains that are there. So these are called as adapter proteins that are present here. So these adapter proteins has the capability to bind to several caspase like caspase 8, and then it undergoes some downstream activation pathways like it activates these proteins caspase 3, caspase 6, caspase 7. These are all caspase that are effector caspases, and whenever these caspase proteins are activated, it has the capability of activating other nucleases and proteases, which helps in the degradation of the nuclear lamina present in the nuclear membrane, and also proteases that cleaves the cytoplasm as well as cytoskeletal protease there. So this leads to membrane blubbing and everything, and also nuclear membrane disintegration, and also DNA degradation and everything. DNA is also activated there, right, because of the activation of nucleases there. And also this individually leads to cell death or apoptosis. So if you look into intrinsic pathway, it can be regulated by this protein called as P53, which is activated by internal stimuli and everything. It can also indirectly activate this particular receptor protein, that is FASL protein or receptor protease there, which activate the external pathway, external stimuli. So it can internal stimuli can also activate other proteins like BIT, NOXA or PUMA proteins. Activation of this BH3 protease, right? These are pro-apoptotic in nature, which involves the activation of this BCL2 proteins as well as inhibition of this BCL2 proteins, inhibition of this BTH2 protein, and the activation of these pro-apoptotic proteins like BAX or BAC. So what BAX are, whenever BCL2 proteins are inactivated, it promotes apoptosis, right? So there's a diagram change there, so it is quite incorrect there. So whenever this is inactivated, apoptosis is activated there. Whenever this is BCL2 is activated, apoptosis is not there. So whenever this BH3 pro-apoptotic proteins activates this effector pro-apoptotic proteins like BAX or BAC, these actually form pores within the mitochondrial membrane there. So within the mitochondria also, because of the calcium accumulation that is taking place there, cytochrome levels are also elevated there, and it leads to a formation of APF there. And this BAX or BAC protein actually helps in the efflux of cytochrome C from the mitochondria into a cytosol, which leads to the formation of this particular structure called as apoptosome, which consists of this caspase 9, cytochrome C, and APAF1 complex or apoptosis promoting activation factor complex there, and which activates this particular caspase like caspase 3, caspase 6, and caspase 7, which in turn activates these nucleases and proteases, which leads to nuclear lamina degradation and also degradation of cytoskeletal elements, which leads to cell death there. So these two pathways, you can see that the extrinsic pathways as well as the intrinsic pathway is actually getting interconnected to each other. So, but the intrinsic pathway is something that happens within the cell whenever any intrinsic factor is present within the cell, activates the apoptosis or tells the cell to undergo apoptosis. While the extrinsic pathway happens because of the presence of any extrinsic signal that is coming from outside, which activates certain receptors that are found there, and it then activates caspase 8, and then it activates effector caspases, etc. Right? So this is how the interconnection happens. However, if you look into intrinsic pathway, you can see that the BCL2 proteins is something that present there, and also you can see that intrinsic pathway is also governed by P53. However, it also regulates the extrinsic pathway to this regulatory activity and all. TNF alpha is actually a receptor present there, and caspase 1 is also is actually involved in this extrinsic pathway rather than the intrinsic pathway. So the answer to this question is BCL2. So I hope I am clear. So anyone has any doubt? P53 can activate both extrinsic as well as intrinsic pathway. Anyone has any doubt? Okay, so if it is clear, so I will just move on to the next question. Coming on to the next question. Which of the following is not a characteristic of necrosis? Loss of membrane integrity, dependence of high ATP level, inflammation or cell lysis. Which of these is not a characteristic of necrosis? Okay, so I am getting option number 2. So you are right. Basically, the answer to the question is dependence of high ATP level. Other factors are actually characteristic of necrosis, loss of membrane integrity, which leads to formation of membrane blubbing there. These blebs actually fuse together to form large complexes that actually leads to cell lysis and which is also governed by inflammation there. So dependence of high ATP level is not actually characteristic of necrosis. So coming on to the next question. A gain of function mutation in which type of gene can contribute to cancer development? Tumor suppressor gene, proto-oncogene, DNA repair gene or apoptotic gene? Which may be the answer. Yeah, I am getting option number 2. Any other option? Anyone has any other response? So yeah, you are right. The answer to this question is proto-oncogene, right? So tumor suppressor genes actually suppress, from the name itself, suppresses the formation of cell division, right? So or cell growth there or tumor growth there. So it actually promotes apoptosis and everything. So P53 we have discussed before is actually a factor. It actually regulates both extrinsic as well as intrinsic pathway, right? So it plays a central role in DNA repair, apoptosis and cell cycle regulation. When DNA damage is detected, P53 can halt the cell cycle to allow for functional repair or trigger apoptosis. It can either enter into senescence or it can trigger apoptosis if the DNA damage is irreparable. Mutations in the TP53 can lead to failure in critical function, increasing the risk of cancer. So whenever a gain of function in case of tumor function suppressor gene happens, is that in case of P53 and everything, it actually prevents the formation of cancer, right? It's becoming more upregulated there. Similarly, another example is BRCA1 and BRCA2, which are involved in breast cancer and everything. And these genes are involved in DNA repair, specifically in repairing double-stranded breaks. And a mutation in this particular gene can increase the risk of breast, ovarian, or cancer due to impaired DNA repair mechanism. So this is because of loss of function activity. Because mutation in this particular gene can reduce its activity, which increase the risk of this particular cancer formation and tumor formation and everything. So in case of tumor suppressor gene, if any gain of function happens, it actually suppresses the activity of tumor formation there, which reduces the cell division for each and every cell there. But if any loss of function happens there, then chances are that tumor will start to appear because it actually suppresses the tumor formation there, right? So if loss of function actually, in case of tumor suppressor gene, actually promotes tumor development there. While in case of proto-oncogenes, like RAS family of proteins, MIC proteins or ERI1 proteins, what happens is that it is involved in cell, most of these are involved in cell cycle progression, cell proliferation and other signal pathways, etc. So whenever a gain of function actually happens in case of a proto-oncogene, that's why it's called as a proto-oncogene, it promotes cancer development. Whenever a gain of function happens, it leads to formation of cancer there. Whenever a loss of function happens in this proto-oncogene, it acts as a tumor suppressor gene there. Like it shows a tumor suppressor activity because the activity of these genes like RAS protein or MIC genes is actually getting lowered there. So that's the like gain of function of mutation in the proto-oncogene can actually lead to development of cancer in the body. So I hope I am clear. In case of DNA repair gene, like if gain of function happens, then it might likely lead to more repair mechanism. In case of apoptosis gene, if any gain of function happen, it leads to more apoptosis and everything, rather than contributing to the cancer development. So I hope I am clear. So anyone has any doubt? Okay, so I think I am clear. So I will just move on to the next question then. Coming on to the next question. Which is the following is a common feature of cancer cell regarding energy production? Increased oxidative phosphorylation, decreased glycolysis, Warburg effect, reduced glucose uptake. Which is a common feature of cancer cell regarding energy production? Yeah, I am getting option number 3. So basically you are right, it's called as Warburg effect, right? Aerobic glycolysis actually gets upregulated there. So what happens is that in case of cancerous cell, large amount of glucose is getting uptaken into a cell, right? Because of that, right, it undergoes rapid glycolysis, large amount of glycolysis in the body. It produces 2 molecules of pyruvate, and whenever in a body, like in a normal cell, the cell prefers to enter into oxidative phosphorylation. What happens is that ATP formation had to take place, right? So in case of cancer cell, it's totally the opposite. So if you look into option number 4, as it takes up most amount of glucose from the body, it increases the glucose uptake, so it is wrong. And it also, because of this higher amount of glucose uptake that is taking place in the body, it leads to increased glycolysis rather than decreased glycolysis. So option number 2 is also wrong. So in case of this cancer cell, instead of entering into oxidative phosphorylation, that is electron transport chain and all into a mitochondria, it undergoes lactic acid fermentation. So it shows it reduced or decreased oxidative phosphorylation. So option number 3 is correct there. So because of the lactic acid fermentation, large amount of lactate is produced there along with a higher amount of energy production, because glycolysis is enhanced there, so large amount of ATP is produced there. Rather than entering into oxidative phosphorylation, it undergoes lactic acid fermentation, leading to large amount of lactate formation. And this lactate is utilized for undergoing metabolism that is taking place in the body there. So aerobic glycolysis is at a high level in this cancer cell, and this factor process called as Warburg effect. And you can see the statement there, like most of the statement that I have said is similar to what is written below, so you can just read it out afterwards. I will share the slides also. So anyone has any doubts in this particular question? Should I explain it once more? Okay, so I think it's clear, there are no doubts. So I will just move on to next question. Okay, so which of these proteins are responsible for membrane blobbing in case of apoptosis and all? CAT proteins, ROC1 protein, BCL2, or all of the above? Which may be the answer? Which of these proteins are responsible for membrane blobbing? Okay, so the answer to this question is ROC1 protein. So this is the pathway. You might have seen this pathway in the lecture videos and all. You can see that caspase 3, once it gets activated, it can activate other proteins like this particular ROC1 protein, which is a Rho activated serine or threonine kinase protein. So it is actually bound to each other. So it consists of a regulatory domain, or consists of a regulatory domain as well as a what is a or a catalytic domain. So whenever it becomes to be active, whenever caspase is activated, this regulatory domain is separated from the catalytic domain, and whenever it becomes active, it has this kinase activity is active. It has a capability to phosphorylate this myosin regulatory light chain there. So whenever this myosin regulatory light chain is there, as it is a part of a cytoskeleton there, right? So it becomes separated from the actin filaments there, and therefore the cytoskeleton gets disintegrated there. So that leads to membrane blubbing there, because membrane blubbing is actually a causative of disassembly of these cytoskeletal elements that are happening within the cell there. So this particular ROC1 protein is actually a it carries out this membrane blubbing formation and everything by phosphorylating this myosin regulatory light chain, which disassembles the cytoskeleton and all. So CAT domain is also called as a CAT protein. The first one is actually a caspase activated DNA free. So it is bound to this inhibitor protein there. So whenever caspase 3 is active, similarly it becomes free from this inhibitor protein there, and this DNase that within the name itself, it has the capability to degrade DNA and cause DNA fragmentation there. So those two are the function. However, in this case, CAT domain actually leads to DNA fragmentation, while ROC1 or Rho activated serine or threonine kinase can carry out membrane blubbing, which is the right answer for this question. While BCL2 is actually an anti-apoptotic protein, right? So it has nothing to do with membrane blubbing. It prevents membrane blubbing there. And all of the above is not the answer, because each and every protein has a different functionality there. So I hope I am clear. So anyone has any doubt? Okay, so I think I am clear. So I will just move on to the next question. Okay, so coming on to the next question. Which of the following is not a characteristic feature of apoptosis? Chromatin condensation and nuclear fragmentation, 4-peptidyl serine exposure on the outer leaflet of plasma membrane, induction of inflammatory response due to cell lysis, and formation of apoptotic bodies. So this is not a characteristic of apoptosis. Yeah, I am getting option number 3. Anyone else? Yeah, so I think each and every one of you agree with option number 3, right? So basically you are right. So induction of inflammatory response due to cell lysis. Actually it's a characteristic of necrosis, right? Inflammation is actually a characteristic of necrosis, not apoptosis. Because apoptosis happens without any painful thing happening to our body. It's actually a characteristic of necrosis. Chromatin condensation and nuclear fragmentation is actually characteristic of apoptosis, because we have earlier discussed with it activates some caspases like DNases and also some proteases that leads to disintegration of nuclear membrane and also DNA fragmentation, right? So from the previous question you can see that CAD is also a caspase active DNase that lead to DNA fragmentation. It is a characteristic of apoptosis. And also 4-peptidyl serine exposure on the outer sleeve set of plasma membrane. So basically it's a lipid molecule there, right? 4-peptidyl serine. So this exposure on the outer leaflet of plasma membrane indicates that the cell is undergoing apoptosis. It needs to undergo apoptosis there. So it actually acts as signaling or a marker that is present at the cell surface there. And the formation of apoptotic body, from the name itself, like the cell actually begins to disintegrate into small bodies. These bodies are also called as apoptosis, the final stages of apoptosis. So that's also a characteristic of apoptosis. So out of this, induction of inflammatory response due to cell lysis is actually not a feature of apoptosis, it's a feature of necrosis. So I hope I am clear. So anyone has any doubt? Okay, so I think it's quite clear. So I will just move on to the next question. Okay, coming on to the next question. What is the primary function of inhibitor of apoptosis protein or IAPs? To activate caspases, to prevent cytochrome C release from mitochondria, to inhibit activate caspase activity and suppress apoptosis, to enhance apoptotic body formation. Inhibitor of apoptosis. Yeah, I'm getting option number 2. Any other option? Yeah, again option number 2. So I think each and every one of you agree with option number 2, right? So, but the option answer to this question is option number 3, to inhibit caspase actives and suppress apoptosis. So basically, these are inhibitors that bind to caspase. Basically, cytochrome C is a different thing. Basically, it releases cytochrome C is involved in mitochondria like apoptosis. It's released from mitochondria, it triggers the activation of caspase, etc. So this inhibitor of apoptosis actually, this protein actually comes as a intermediate between this particular pathway that it cytochrome release and the activation of this caspase, etc. So whenever this happens, like whenever these proteins are present, these bind to these caspases, etc., and it has to degrade them out, including caspase 3 and caspase 9, whichever is involved. So these inhibitor proteins bind to these proteins, and then it activates this particular pathway which inhibits the apoptosis and all. So option number 1 is not right because it inhibits caspase, not activities. It prevents cytochrome C release from mitochondria. No, it's actually a function of other proteins. Option number 3 is the right answer that we are discussing, and option number 4, to enhance apoptotic body formation. Basically, it inhibits apoptosis, so it cannot enhance the apoptosis body formation. It reduces the amount of apoptosis body that is getting formed there. So that's also a wrong answer. So inhibitor of apoptosis protein actually inhibits apoptosis by binding to it and degrading it out, and then suppresses apoptosis. So I hope I am clear. So anyone has any doubt? Okay, so I think I am clear. So so I will just move on to the next question then. So coming on to the next question, which factor is an important for angiogenesis in cancer cell? Epidermal growth factor, vascular endothelial growth factor, insulin-like growth factor, fibroblast growth factor. Which might be the answer? Okay, I am getting option number 2. So basically you are right. The answer to this question is vascular endothelial growth factor or VGF growth factors. So yeah, so this is just a short diagram basically. So you can see that the this is the cancerous growth there, right? So it is present at this particular part of the tissue. So what it means is that it secretes out several growth factors like VGF, EGF, fibroblast growth factor, TGF, and IGF-1, which has different functions and all. So it binds to this receptor that is present within the blood vessels and all. So whenever this happens, the vascular development starts to happen. You can see that cell recruitment is happening there, and then the cell blood vessels start to form, and it then interacts with this cancerous cells there. So whenever this happens, this is initiated due to the activity of VGF or vascular endothelial growth factors there. So this leads to vascularization, and all the nutrients that the cell require happens because of this vascularization and all. So that's how tumor cell has the capability to induce blood vessel formation by a process called vascularization, which is characterized by this VGF. And this vascular or blood vessel development is one of the main ingredient that leads to cancer cell proliferation and survival, etc. So and it is one of the important process of angiogenesis. So yeah, so answer to this question is VGF or vascular endothelial growth factors there. So anyone has any doubt regarding this? Okay, so I think it's clear, so I will just move on to the next question. Coming on to the next question, in which phase of the cell cycle are senescent cells occupied at? G1 phase, G2, G0 or S phase? Okay, so the answer to this question is G0 or quiescent phase, right? So it's actually is correlated with the G1 phase. So you can see that there's a restriction checkpoint there. So this restriction checkpoint prevents the entry from G1 phase to G0 S phase there. So whenever cell cycle arrest needs to happen, the cells that are present in the G1 actually enter into a G0 phase, which actually gets arrested at this stage, and this promotes the senescence phase of the cell cycle there without entering into the cell division that you have discussed before, right? So yeah, so in this phase of cell cycle, that is the G0 phase of cell cycle, the senescent cells are occupied. So anyone has any doubt regarding this? Okay, so I think it's clear. So I will just move on to the next question. Which of these is a marker for cell senescence? P10, P15, P16 or P20? Like there are several proteins like P53 and everything, right? So there is a specific protein which is actually acting as a marker that this cell is undergoing senescence. Which one of these is that marker? Okay, I am getting option number 4. So the answer to this question is P16. So basically you can see that this is a particular diagram there. So it can either activate P53, which activates P21, which is interacting with a retinoblastoma, right? So P16 is also there. It inhibits certain CDKs that actually phosphorylate retinoblastoma there. So whenever is there, the cells signal that whenever the retinoblastoma is activated there, it has the capability to signal the cells that the cells should enter into a phase called a senescence phase, that is enter into a G0 phase of the cell cycle from the G1 phase. And also because these cells are not retinoblastoma is not for getting phosphorylated, it is active, it is bound to ETOF, right? So whenever it is bound to ETOF, it cannot synthesize some other genes, or it cannot act as a transcription factors to regulate the synthesis of other genes. that are promoting the what you say cell division there. So retinoblastoma is still bound to this E2F gene, so the cell gets arrested into a G0 phase there rather than entering into a G1 to S phase or passing the G1 checkpoint there. So that's what P16 does. So P16 actually inhibits this CDKs that phosphorylate this retinoblastoma cells there, retinoblastoma protein there, which in turn gets bound to E2F, which in turn promotes inhibits the synthesis of this particular transcription factors there. So P16 is actually a marker for cell senescence, and in turn promotes senescence there. So anyone has any doubt? Okay, so I think it's quite clear. So I will just move on to the next question. So coming on to the next question. Mitochondria has the capability to regulate cell aging. Which of the following statement is incorrect? Defective quality control by mitophagy, altered mitochondrial fusion or fission dynamics, in DNA or nuclear DNA mutations and increased mitochondriogenesis, stabilization of ETC complexes, which might be the answer. Which of these following is incorrect regarding how the mitochondria regulate cell aging? Okay, so I think I will just give you the answer there. So answer to the question is in nuclear DNA mutation and increased mitochondriogenesis. It should be reduced mitochondriogenesis, because in order to regulate aging and all, the mitochondrial levels goes down with the cell in an aged cell. So it does not increase mitochondriogenesis, it reduces mitochondriogenesis. Other options are correct. Defective quality control by mitophagy. Mitophagy doesn't happen, so quality control is not maintained there because of cell aging. Altered mitochondrial fusion or fission dynamics is also a part of cell aging. Nuclear DNA mutations do happens in this case, but a reduced mitochondriogenesis happens in this case. So this statement is wrong. Destabilization of ETC complex do happen, leading to reduced ATP production and all, which in turn leads to cell aging. So I hope it's clear. So anyone has any doubt? Okay, so I think it's quite clear. I will go to the last question then. So this is just a diagram. We have discussed this before. Mitochondrial mutation lead to mitochondrial diffraction, defective mitophagy, altered fusion or fission, destabilization of ETF complex, nuclear damage leading to reduction in this particular phosphoglycerate cyclase enzyme, leads to reduced mitochondriogenesis and mitochondrial diffraction, which in turn leads to aging by reducing the bioenergetics, increased cell death and inflammation. So I will just move on to the next question then. Last question. Which of the following pathway is also called as a death receptor pathway? Extrinsic apoptotic pathway, intrinsic apoptotic pathway, senescence or necrosis? Which might be the answer? Yeah, I'm getting that. So extrinsic apoptosis pathway, this is apoptosis pathway, right? So receptors are present there. The presence of this caspase AC gets activated, and this can then activates caspase 8 and other caspase leading to chromatin condensation, DNA fragmentation, membrane blubbing, phagocytosis, etc., which is characteristics of apoptosis. So I hope I'm clear. So anyone has any doubt? Okay, so I think it's clear. So by this we can end the section for today. So thank you everyone for joining. So if does anyone has any doubt regarding the like what you say, the whole meeting that you have done today, or regarding the question? Okay, so I think if there are no questions, I think we can end the section for today. So thank you for joining. So I hope that everyone else will join for the other class also, for the other section. Thank you.